Okay, class. Good day. So for today's meeting, we will tackle your lesson for the complement system. Now, since module one, you've been hearing the complement system over and over again, which is part of the adaptive immunity. Aside from antibodies, this would also include the adaptive immunity. Now, the complement system class would be a composition of different proteins that would aid in fighting against the certain antigens. So let us begin. Now, please take note, class, that the complement system, as earlier mentioned, is made up of complement proteins. Now, what are these proteins, class? This is a group of more than 30 circulating and membrane bound regulatory and constitutive component glycoproteins that have different functions. They would function to promote host defense against infectious agent. They promote the clearance of apoptotic cell debris and immune complexes. They regulate the immune response. So please take note of that. They promote host defense against infectious agent, clearance of apoptotic cell debris and immune complexes, and they help regulate the immune response. Now, please take note, class, that the complement system of proteins is phylogenetically older than immunoglobulins and is present in primitive organisms that would lack antibodies. It states here, class, that the complement system was already present before antibodies were even created. They were somehow one of the original defense system of the body, and can be found in primitive organisms that have no antibodies. Now, please take note, class, that in the complement system, there are three pathways. Now, what are these three pathways? We have number one, the classical pathway, number two, the alternative pathway, and number three, the MANO MBL or the Manos binding lectin pathway. Now, for every pathway, class, for every pathway, there is a corresponding group of complement proteins. An example of that is that for the classical pathway, class, this is composed of nine proteins, nine complement proteins. So please take note of that. If asked in your exam, how many complement proteins are there in your classical pathway? There are nine. They are from your C1 to C9. Now, aside from C1 class, C1 is subdivided into three types. They have the so-called C1QRS complex. And for every corresponding letter, they have a function. Let's tackle your C1Q. Now, you don't need to memorize the molecular weight, the concentration, class. I just want you to take note of what the name of the complement protein and their function. So let's start with your C1Q. Your C1Q class would bind to the FC region of IgM and IgG body. Now, if you still remember the appearance of your antibody, they are typically Y-shaped, and they are composed of two parts: the Fab and the FC regions. Now, the Fab region would be here, and the FC region would be here. Now, it states here that the C1Q would bind to the FC region of IgM. So, C1Q here, class. C1Q would bind to the FC region of the IgM and IgG antibody. Now, after C1Q has attached, class, C1R would now come. Now, C1R would come and it would activate C1S. With C1S arriving, it would now cleave your C4 and C2. So later I will explain that. So let me repeat, class. C1Q would first bind to the FC region of your antibody, specifically IgM and IgG. Follow that, class, C1R will arrive and it would activate C1S. C1S would now cleave C4 and C2. Now, your C4 here, class, please take note, is a part of your C3 convertase, specifically the C4b. Now, each complement protein class, please take note of this. Each complement protein class can be cleaved or can be cut into two fragments. And they are characterized by the letters A and B. For example class, if you cleave or cut C4, it would form C4A and C4B. Now it mentions here that the C4 is part of your C3 convertase, specifically the C4B fragment. So I hope that was clear. So if asked in your exam what is the purpose of your C4, it's part of the C3 convertase, specifically the C4B. Now for C2, it binds to C4B, forming your C3 convertase. So later I'll explain that again. Just take note of the function. And for the C3, this is the key intermediate in all pathways. Now when you say key intermediate in all pathways, it is the one that is common. It is the one that is common to all pathways class. This is the one common to all pathways. Then your C5 would initiate the so-called membrane attack complex. Now, the membrane attack complex is known also as your MAC. And the MAC class is responsible for lysing the pathogen or the antigen. So later, I will again explain that in the coming slide. Your C6 class would bind to C5b in MAC. C7 would bind to C5b, C6 in MAC. C8 would start pore formation on the membrane, and C9 would polymerize to cause cell lysis. So later I will better explain class this classical pathway. Then we also have the second pathway, the alternative pathway. Now in the alternative pathway class, this is composed of three complement proteins. They are number one, factor B, factor D, and proper D. Now factor B class would bind to C3b to form C3. Factor D would cleave factor B. Properdine would stabilize the C3b, BB, C3 convertase. Then for the MBL pathway, you have your MBL. This is the mannose binding lectin, which would bind to the mannose. MAS1 has an unknown function. MAS2 would cleave C4 and C2. So do try to memorize this table class, excluding the molecular weight and the concentration. They might be included in your exam and in your coming quiz. So let's continue. Here's a review question. How many complement proteins are involved in the classical fat? So the answer to this is that there are 9 complement proteins, C1 to C9. And your C1 class is composed of a C1q RS complex. Let's continue. And let's talk about the nomenclature, or how do you typically write the complement proteins. Start with the classical pathway nomenclature. Now, the 9 proteins of the classical pathway are designed class by the uppercase letter C, which is then followed by the number that relates to their order of appearance in the reaction sequence. Note is that a notable exception here is C4, which would act C2. Now, normally class, if you follow the rule of thumb in following the order of numbers, it would be 1 to 9. But in the complement, in the classical pathway, what happens is that the first complement would be C1, followed by C4, followed by C3, C2. Sorry. It should be C1, C4, C2, C3, C5, C6, 7, 8, and 9. So that's the order of appearance of your classical pathway. So it should start with C1, followed by C4, C2, C3, 5, 6, 7, 8, and 9. So I hope that was clear. And when writing it, you would use the uppercase letter C, followed by the number of their order of appearance. fragments. Now, molecules that are part of the C1 complex are C1q, C1r, and C1s. So as I mentioned, class, your C1 is made up of a complex of C1q, R, and S. Now, for the alternative pathway class, please take note that these are proteins reacting slowly in the alternative pathway, and they are called factors. So, if the classical pathway class, for the classical pathway, you're going to use the uppercase letter C, followed by the number. For the alternative pathway class, you're going to use factors, followed by an uppercase letter. So we have your 2, factor B, and factor D. So I hope that was clear. So for the alternative pathway, you would use the word factor, followed by the letter B, big letter B, and big letter D. Now for the MBL, or the mannose binding lectin pathway class, this is a pathway designated by the abbreviations of the names of proteins. So this time we will use abbreviation. And there are only two abbreviations here that you need to remember, class. MBL and MASP. MBL would stand for Mannan binding lectin or mannose binding lectin, while MASP would stand for MBL associated serine protease. So just a recap, let me repeat. If it's the classical pathway, you would write it by using the capital or the big letter C followed by the number of the order of their appearance. So C followed by the number. For the alternative pathway, you would use the word factor followed by the big letters B, factor B, or factor D. And then for the MBL pathway, you're just going to memorize the abbreviations MBL and MAS. MAS would stand for, again, MBL associated serine protease. MBL would stand for Mannan binding lectin. Nomenclature. Please take note that fragments from cleavage of complement proteins are, okay, as I mentioned earlier, class, in the table, your complement proteins class could be cleave or cut. Now, if they are cleave or cut, they would form two fragments. So let's say you cleave your C3. If your C3 is cleaved, class, it would form C3A and C3B. Now, what does this A and B stand for? Please take note, class, that the A here would stand for the smaller fragment. So imagine this would be your C3. Mari lang ganyan yung C3. C3 would now be, and if you try to cleave it, the smaller fragment would be the letter A, while the larger fragment would be the letter B. So I hope that was clear. So if asked in your exam, what letter would stand for the smaller fragment of a cleaved protein, cleaved complement protein, the answer is A. If asked in your exam, what letter would stand for the larger fragment of your complement protein, it would be your B. Now there is an exception or exemption to this rule, class, which is C2. C2 is the only one that is opposite, class, wherein C2A is the larger fragment and C2B is the smaller fragment. So aside from the rest of the complement protein class, A would be the smaller, B would be the larger, but again there would be an exception, which is your C2. In C2A class, this one would be the larger fragment, and for C2B, it would be the smaller fragment. So do take note of that. Now here's a review question, class. Which complement protein is the exemption in regards to the smaller and larger fragment? So the answer to this is your complement protein C2, wherein C2A class would be the larger fragment, while C2B would be the smaller fragment. So let's continue. Let's tackle this time, class, the complement pathway. Let's start with your classical pathway class. Now, the classical pathway class is the first pathway to be studied. Now, thus, it was called the classical. Uh, Kaya siya tinawag na classical class because it was the first pathway to be recognized, to be studied. Now, this is responsible class for complement activation on most antibody sensitized cell. Now, whenever you would hear antibody sensitized cell class, we are talking about your antibody antigen complex or binding. An example of this class, let's say, if you still remember in the pictures in your module 1, this would be your bacterial cell. If you still remember in the bacterial cell class, on its surface would be epitopes or determinant cell. So imagine this would be the epitope, this triangle. Now your antibodies would arrive, and if you still remember, the antibodies have the Fab site or the paratope. So what happens is that the Fab site or the paratope would attach itself to the epitope, forming the so-called antibody antigen complex. Now, please take note that for your classical pathway to be activated, it would require this so-called antibody antigen complex or antibody sensitized cell. Now, they are made up of 9 number proteins, C1 to C9. Activation of the classical pathway would usually require the interaction between antigen and a C1 binding complementing antibody. Now, whenever you would deal with C1 binding complement antibody class, this would refer to 2 types of antibody, the IgM and the IgG. So these are the only antibodies class capable of activating your classical pathway. So let me repeat, if asked in your quiz or exam, what activates the classical pathway, it would be your antibody antigen complex or antibody antigen interaction or antibody synthesized cell. And if asked what are the 2 antibodies that are C1 binding, the answer is your IgM and your IgG. Now, these are the summary class of what I mentioned. These are the immunoglobulins that can activate the classical pathway. We have your, as I mentioned, IgM and IgG. Now, the thing you need to remember, class, about your IgG is that it has 4 subclasses. It has 4 subclasses which we will tackle in Module 6. They are your IgG1, IgG2, IgG3, and your IgG4. Now, please take note that only IgG1, 2, and 3 are capable of activating the classical pathway. So let me repeat. It is IgM, IgG1, 2, and 3. These 4 antibodies are capable of activating the classical pathway, while IgG4 class is not capable of activating the classical pathway. That is why if asked in your exam which IgG subclass is not capable of causing or activating your complement pathway, the answer would be IgG4, as well as the other antibodies A, E, and D. So I hope that was clear. So let me repeat. IgM, IgG1, 2, and 3 are the immunoglobulins capable of activating the classical pathway, while IgG4 is the only immunoglobulin G subclass not capable of activating the complement pathway. They would include IgA, IgE, and IgD as well. So please take note of that. So this is the process class on how you would activate the classical pathway. Now, If you take a look at the first picture on the left, this would be class. This would be the bacterial surface, or the bacteria, or the pathogen, or the antigen. Now, if you take a look at the picture, you would see the antibodies. So, ito yung antibodies class. These are the antibodies. Now, what happens here is that once there is the antibody antigen complex caused by either IgG 1 to 3 or IgM, this will now cause class the arrival of the complement system, or the classical pathway. This is your C1QRS. Now, when C1QRS class arrives, specifically your C1S class would cleave C2 into 2. We have your C2A, C2B. It would also cleave class C4, forming C4A and C4B. Now, what happens in this part class is that the C4B, the C4B class would separate on its own, and it would now cause C2A to attach, forming your C4B2A. Now, C4B2A class is now known as the C3 convertase. Now, from the word C3 convertase, it would now act on C3. It would now act on C3, cleaving C3 into C3A and C3B, forming your so-called C4B2A. This time, the 3B, the C3B will bind with C4B2A, forming your C4B2A3B, also known as the C5 convertase. Now, the C5 convertase class, please take note of that, would act on C5, cleaving it into 2, C5A, C5B. Now, what happens is that C5B would separate, and it would cause the attachment of C6 to 9. So, it would now form your C5B, 6, 7, 8, and 9, which is now known as the membrane attack complex. And it would lyse the antigen. So, para mas maintindihan nyo, let me just show you a diagram. So, I do apologize for the drawing class. So, let's start from the top. Now, whenever there would be an antibody antigen complex, it would cause class the arrival of your C1QRS. Now, what C1QRS does is that it would cleave two complement proteins. It would cleave your C4, forming C4A and C4B. It would also cleave C2, forming C2A and C2B. Now, what happens here is that C4B, which is separated, will cause C2A to attach to it, forming C4B2A, which is now known as the C3 convertase. Now, C4B2A would now attack, or would now cleave C3, forming C3A and C3B. Now, what happens next is that your 3B would attach to your C4B2A, forming your so-called C4B2A 3B, which is now known class as the C5 convertase. And that C5 convertase, C4B2A3B, would now cleave C5, forming C5A, C5B. Now, what happens next is that we have now your C4B2A3B. C5B class would now cause the binding of C6, 7, 8, and 9, forming your membrane attack complex, which would cause the destruction or the lysis of the antigen. So that's the classical path. So let me repeat, class. Whenever there would be an antibody antigen complex, C1QRS would arrive and it would cleave C4 and C2, forming C4A, C4B, C2A, C2B. C4B would cause the attachment of C2A, forming your C4B2A, known as the C3 convertase. Now, it would now cleave C3, forming C3A, C3B. What happens, class, is that C3B will attach to C4B2A, forming your C4B2A3B, known as the C5 convertase. The C5 would now be cleaved by C4B2A3B, forming C5A, C5B, and C5B would cause the attachment of C6, 7, 8, and 9, forming the membrane attack complex, leading to the lysis of antigen. That is why if it is asked in your exam class, what is the C3 convertase of your classical pathway, the answer would be C4B2A. Pag tinanong naman, what is the C5 convertase? It would be C4B2A3B. So please do take note of that, class. Pag tinanong kayo sa exam, what is the MAC of your classical pathway? The answer would be C5B, 6, 7, 8, and 9. So I hope that was clear. All right, here's a question. What is required for the classical pathway to be activated? So the answer to this question, class, is that they will require an antibody antigen binding complex or interaction. Let's continue. Let's go down this time, class, to the mannose binding lectin pathway. Now, the difference, the thing you need to remember about the MBL pathway is that it is almost similar. The only difference, class, is how it is activated. How it is activated. So this is similar with your classical pathway, but difference on how it is activated. So instead of activation through antibody binding, the lectin pathway is activated by recognition of surface moieties that are found on certain pathogens. So this time, class, there is no antibody binding, but instead, it would require the presence of certain moieties or substances found on pathogens. And one of those substances, class, is the so-called mannose. And this is usually found in the carbohydrate. This is a carbohydrate class found in the cell wall, found in the cell wall of your bacteria. Now, this pathway, class, would provide an additional link between innate and acquired immune response because it would involve non-specific recognition. Excuse me. Remember, this doesn't use, this does not involve antibodies, class. Therefore, if you still remember, di ba, ang antibodies are usually involved in specific immune response. But due to the help of this MBL pathway, it makes a connection between the innate and the acquired. So your antibody is part of the acquired immune system, di ba? With that, class, this serves as a linkage between the innate and acquired immune response because it would involve non-specific recognition. As long as there is the presence of mannose, class, or other moieties found on the microbial cell wall, they could easily activate. So I hope that was clear. Now, the lectin pathway molecules are structurally similar to those of the classical, The classical and lectin pathways even share the same components C4 and C2. And once C4 and C2 are cleaved, the rest of the pathway is identical to the classical pathway. The role C1q would serve in the classical pathway is filled by three classes of recognition molecules in the lectin pathway. So uh, instead of having C1q class, there is no C1q here to recruit your R, your S. Here, class, there would be lectins, ficolins, and CLK1. So let me repeat, instead of C1q, which is found in the classical pathway, for the MBL pathway, there would be lectins, ficolins, and your CLK1. So let's have the diagram. Dito muna tayo. All right. This is your mannose binding lectin class. This one. Itong malaking to. This is the mannose binding lectin. Now, whenever it would find your mannose, it would cause the arrival of the lectin, the ficolin, and the CLK. Now, what happens here is that that happens, class, it would cause the cleavage of your C4 and your C2, forming again C4A, C4B, C2A, C2B. Now, again, from the top, it will cause your C4B to form your C4B2A, known as the C3 convertase. And it would cleave C3 into C3A, C3B, forming your C3A, C3B, and C3B will attach to C4B2A, forming your C4B2A, 3B, also known as the C5 convertase. Now, the C5 convertase would now act on C5, forming C5A and C5B. Now, C5B class would now further be attached with your 6, 7, 8, and 9. Then that would lead to the membrane attack complex, causing the destruction of your bacterial surface. So if you notice, class, similar lang sila. They are similar. They are very similar with your classical pathway. The only difference is that the way it is activated. So it would be activated by certain carbohydrates found on the bacterial surface, such as your mannose. Balik tayo dito. Okay. So whenever it would detect certain carbohydrates, class, such as your mannose, on the bacterial surface, it would cause the release of your lectins, your ficolins, and so on. Causing the cleavage of your C4 and your C2. Now, C4 would become C4A, C4B, C2 would become C2A, and C2B. Now, C4B again will attach with your 2A or C2A, forming the C4B2A, known as the C3 convertase. Now, this would act on C3, forming your C3A and C3B. Now, C4B2A, now C3B would attach to C4B2A, forming your C4B2A, 3B. This is now known as your C5 convertase. Now, the C5 convertase C4B2A, 3B would now cleave C5, forming C5A, C5B. C5B class would now cause the attachment of C6, 7, 8, and 9, forming the membrane attack complex, lysing the bacterial cell. So do take note of that. So it's similar class with your classical pathway. The only difference is that it would be different in the how it is activated. All right, let's go to the last pathway class, the alternative pathway. This was originally named class for the protein properdin. This is a constituent of normal serum class. It functions mainly as an amplification loop for the activation started from the classical or lectin pathway. So this is somewhat class an enhancer. It enhances the classical and lectin pathways. Now, although properdin has been confirmed to bind and initiate activation, the primary function of properdin is to stabilize the C3 convertase formed from activation of other factors. So if asked in your exam what is the primary function of properdin, it is to stabilize your C3 convertase. In addition to properdin, the serum proteins factor B and D are unique to this pathway. Ito yung pathway niya, class. So the alternative pathway, the difference class is it would start, as mentioned earlier, this is an amplification loop. Now this would start class with your C3. Now what happens here class is that C3 would be hydrolyzed. When you say hydrolyzed, it would be cleaved. It would now become C3A and C3B. Now we don't care about C3A, let's just focus with C3B. Now what happens with C3B class is that it would attach to your factor B. It would attach to your factor B, and when it is attached to your factor B class, factor B now class would now be cleaved. It would now be cleaved by factor D. And again, pag cleave siya class, it would form your BA, BB. Now, si BA, wala ulit tayong pakialam. Since originally class, C3B was already attached to factor B, it would now form class your C3B, B, B. Now, C3B, BB class is now known as your C3 convertase. Now again, since this is a C3 convertase class, it would cleave more C3. So magkikleave na naman siya ng C3 class. So it would cleave your C3 forming your C3A, C3B. Now what happens is that the excess C3 class, the excess C3 would now attach to the C3B BB, now forming the so-called C3B BB 3B. And this would now be known class as your C5 convertase. Now, earlier class it was mentioned that properdin would stabilize your C3. Now, when properdin would usually attach here after the BB. So forming your C3B BBP. Now, if you continue this with the C5 convertase, it would read as C3B BBP 3B. So that would now be your C5 convertase. Now the C5 convertase C3B BBP 3B would now act on C5, cleaving C5A to C5B, and C5B would again cause the attachment of 6, 7, 8, and 9, forming the MAC and leading now to the lysis. So para mas maintindihan nyo, for your better understanding, let's do this on a diagram. Kuha tayo ng isang excess slide. Okay. So we have the alternative pathway. So the difference between your alternative pathway class is that it would begin with your C3. Now C3 class would be cleaved by water into C3A and C3B. Now C3B here class would now attach with your factor B. And factor B class would be cleave. Iki-cleave yan ni factor T. Now, when it is cleave class, when in your factor B is cleave, it would form your BA and your BB. Now, your BA class would be removed and what will remain is BB. Now, what happens is that the originally attached C3, yung C3B yung may iwan, yung larger fragment, C3B would cause the attachment of the BB fragment. So, it would now form the C3B BB. Now, this is known as your C3 convertase. Now, if you still remember in this pathway, very unique si properdine class. Properdine would stabilize the C3 convertase. So, you would now add your properdine, C3B, BB, P. Now, as you all know, this is the C3 convertase. It would now continue to act on more C3, cleaving C3 into C3A and C3B. Now, C3B here class would now attach to the C3B BBP, forming the C3B BBP 3B. So you would now have your C3B BBP 3B. And this would now be known as the C5 convertase, which would cleave C5 into C5A, C5B. And C5B class would now attach to 6, 7, 8, and 9, forming the MAC, which would lyse the antigen or the pathogen. So here's a question dito, class. Pag tinanong kayo sa quiz or exam, if you were asked in the quiz or exam, what is the C3 convertase of your alternative pathway? The answer would be your C3B BB P. Pag tinanong kayo sa exam, what is the C5 convertase? It would be C3B BB P 3B. And pag tinanong kayo what is the MAC of the alternative pathway, same lang din. C5B, 6, 7, 8, and 9. So I hope that was understandable. If you have questions, type them in the chat box. So let's continue. Okay, here's a review question, class. Aside from your factor B and factor D, what is unique to the alternative pathway? So the answer to this, class, is the protein property, which would stabilize your C3. Okay, let's continue. Now, C3 is a key component of all three pathways, class. Now, C3 is a molecule synthesized in many cells, particularly hepatocytes, and is released into the circulation, where it would bind to foreign target surfaces. The activation of and conversion of C3 to C3b leads to covalent binding C3b to the surface of a target, such as the microorganism. The target would become coated with C3b, capable of interacting with cells expressing C3b receptor, including all phagocytes, all B lymphocyte, and a subset of T cell. Now, para mas maintindihan nyo to, class, ito. This is the classical and lectin pathway. This is the alternative pathway. Now, if you try taking a look, so this is how it starts. So it would start for the classical, yung C1QRS. For the lectin pathway or the MBL pathway, it could be the mass starter. Now, if you take a look at at this class, all of the pathways, even the alternative pathway class, they would all meet with C3. C3 class is the key component of all your pathways. That is why if C3 complement protein is absent class, if this is absent, there is no complement activation, or rather, not complement activation. No successful complement system. So I hope that explains it. So this is again the summary of what I explained earlier. So that's your classical pathways, you went URS, your MBL, the MAS, they would attach to the Manos. Then for the alternative pathway class, they would usually be activated in the presence of certain substances of your bacteria, such as teichoic acid, chymosan, and lipopolysaccharide. But this time class, they would begin with the C3. So that's the difference. If classical and alternative would begin with C4 and C2, C alternative niya would begin with C3. Then this is your MAC, the MAC. 5B, 6, 7, 8, and 9. Now let's talk about the membrane attack complex. So the membrane attack complex is the presence of C9. Please take note of this class. C9 class would greatly speed up the lysis. However, sufficient perturbation of the membrane can occur in the absence of C9, so that deficiencies in C9 would appear largely benign. Now, to simplify this class, sabi dito, C9 would enhance the speed of lysis, but technically, C9 is not necessarily 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 required. That is why if the person has C9 deficiencies, it would appear as benign class. Now, C5b678 can lyse unnucleated cells, while you would require 9C9 to lyse nucleated cells. So please take note of that. So that's your MAC. Then summary of the three pathways. So classical pathway activated by IgM and IgG, except IgG4. Starts with C1 activation, C3 convertase C4b2A, C5 convertase C4b2A 3b. MAC is C5, 6, 7, 8, and 9. Then for the alternative pathway activated by polysaccharide and lipopolysaccharide. Start with C3 activation. C3 convertase is C3BBP, can include the P class all the time. In some reference kasi hindi nila sinasama, but just to make it uniform, isama na lang natin yung P. Then the C5 convertase is your C3BBB P3B. The MAC is 5, 6, 7, 8, 9. For the MBL class, starts with the activation of Manos. Very similar to the classical pathway. MBL is structurally similar to your C1Q. And MASP2 is homologous to C1S. So do try to memorize this summary. Then let's go to your anaphylatoxins. Now, what are these anaphylatoxins class? These are fragments that are enzymatically released during complement activation, and they can cause increased vascular permeability, contraction of smooth muscle, and the release of histamine from basophil and muscle. Now, whenever you would deal with anaphylatoxins class, they would tend to resemble an allergic reaction. The reason for that class is it involves histamine from basophil and muscles. Now, although all three anaphylatoxins show bioactivity, relative potency is like this. So the greatest potency class. Please remember this. The greatest to the least. So C5 is greater than 3A, 3A is greater than 4A. So the greatest would be C5A, while the least greatest is your C4A. So do remember that in terms of their potency. C5A followed by 3 and 4. Now they have different functions. So we have C5A, they have a receptor C5AR. This is capable of causing neutrophils to release hydrolytic enzyme, oxygen radicals, and prostaglandin, which would aid in the destruction of foreign antigens. Now, chemotaxin for neutrophils, basophils, eosinophils, mast cells, monocyte, and dendritic cell. Then we have your anaphylatoxin C3A. The receptor is your C3AR, selectively effective in eosinophil localization to allergic site. Then we have your C4A, unknown ang receptor niya. It causes increased stress fiber formation and enhanced endothelial permeability. Now they have a common function class. Please do memorize this. They attach to specific receptors on neutrophil, basophil, mast cell, eosinophil, smooth muscles, and the vascular endothelium. Then let's talk about how your complement is controlled or regulated. So for the complement to be regulated class, there are certain regulatory proteins. So I want you to memorize this class, this table. So we have your C1 inhibitor, dissociates the C1 complex by binding to C1R and C1S. Factor H, cofactor for C3b inactivation by factor 1. C4 binding protein, cofactor for C4b inactivation by factor 1. S protein, vitronectin, inhibits insertion of the MAC into cell membrane. Clusterin inhibits insertion of the MAC into cell membrane. And then factor 1 inhibits C1 complex formation, inhibits cleavage of C3 by BB. Then we also have your CARP or your cell-associated regulatory protein. This would regulate complement activation, which would occur on host cell surface to limit inadvertent injury to those cells. Many of these regulatory proteins would act as complement receptor. So we have your CR1 or your complement receptor type 1. Dissociation of C3, C5 convertase. Cofactor for C3b and C4b. Inactivation by factor 1. Then we have your decay accelerating factor or your DAF, also known as CD55. This is responsible for the dissociation of C3, C5 convertases. Then we have your membrane cofactor protein, MCP. This is known as your CD46, cofactor for C3b, inactivation by factor 1. Then we have your protectin, CD59, inhibits formation of the MAC. Then we have your homologous restriction factor, inhibition of formation of the MAC. Then deficiencies and their associated diseases class. So I want you to memorize this class. I won't tackle them. Just memorize the protein and their major clinical correlations. So most of the correlations naman class are repetitive. May iba lang na naiiba. Tandaan nyo lang kapag C1, glomerulonephritis saka SLE. So do take the time to study and memorize this. This may come out in your exam. Now, the most serious deficiency class would always be the C3 deficiency. Why? Again, class, diba, the C3 is the key component of all your pathways. Kung walang C3 class, hindi matatapos yung complement activation niya. Well, the most common deficiency is the C2 deficiency. Even if your C2 deficiency class is absent, somewhat the complement pathway will still continue. But it would be a weak pathway. So that basically ends the module 4 class, the complement system.